And I'm going to start off with molecular genetics with the notion of the central dogma. Now, the central dogma is this entire process of how genetic information is passed on from offspring to offspring. And this can be explained by looking at this process of DNA going to mRNA, messenger RNA, and then finally or eventually protein. And within these things, various things occur in which we will learn all of them. Over here, DNA has to undergo replication. When we go from DNA to mRNA, we call this transcription. And this messenger RNA finally going into protein is called translation. This whole notion of translating proteins from transcribed mRNAs is referred to as the entire central dogma of biology. This is really where the bulk of biology comes in. So if you haven't been paying attention to any of the videos, I suggest you at least pay attention to the central dogma of biology, or at least molecular genetics, because this is really where the fun stuff comes in. Now, DNA, we all know DNA is now the genetic material of all living things. We've learned it since, third grade? We see it in our textbooks. DNA, DNA, DNA. Some of us know what it stands for, right? Deoxyribonucleic acid. But what you may not have known is that just decades ago, this DNA was not considered the genetic material of all living things. People actually believed that protein was the genetic material. They believed that protein was being passed on, not DNA. So now we're going to talk about a set of experiments that really set the understandings for understanding the whole field of molecular genetics. And we're going to first talk about Hershey and Chase. These guys carried out a very, very, very simple experiment, but a very, very intelligent one, with intelligent design. Hershey and Chase. So there was an entire debate going on. Protein or DNA? Protein or DNA? What's passed on? So they say, hey, we're going to solve this problem. Let's see how to do it. So they took bacteriophages. And of course, inside is my genetic material, which in this case is DNA. And they had a bacterium. And of course, this bacteriophage is going to ultimately infect this bacterium. And you would you would hope to see more bacteriophages growing inside the bacterium. It's supposed to infect this bacterium, and you're supposed to see more and more bacteriophages. But what's the element? What is the substance that's being passed on here? Is it the protein? Is it the DNA? So now let's look at a bit of background of this experiment right here. Here's my bacteriophage, and this entire thing is basically protein. DNA is not protein, remember that. Okay, that's a very big misconception. Some people get confused all the time about it. This right here is the protein. Inside is the DNA. Proteins are made up of specific elements. DNA are made up of specific elements. And they said, hey, why don't we radioactively label one of the elements of sulfur that is not present on DNA? We're going to make up two different experiments. We're going to radioactively label an element that can detect for the presence of protein here. And for experiment 2, we're going to radioactively label an element that can detect for the presence of DNA. And that's exactly what they did. They said, okay, you know what? I know sulfur is present only in this protein complex. So sulfur, we're going to use the isotope 35. And they radioactively labeled this protein coat, the bacteriophage, and now what? Bacteriophage is going to infect this bacterium. Here's my bacterium. And if indeed protein is the genetic material, we should also see radioactivity within inside this bacterium. But what did they see? They saw no radioactivity, but they did of course see more bacteriophages growing inside this bacterium. So bacteriophages were growing, but they did not see any sort of radioactivity. Alright, hey, protein is not the genetic material. Okay, alright, fair enough. But is DNA the genetic material? Well, let's see. They set up a second experiment. What did they do? Similarly, they took bacteriophages, here is my bacterium, and they said, you know what, alright, this time we're going to radioactively label an element that is only present on DNA. And they decided to radioactively label the element phosphorus. Phosphorus is present on DNA, and they used the radioactively labeled isotope 32 phosphorus, and now this DNA is radioactively labeled. And the bacteriophage will essentially infect this bacterium, and lo and behold, what do you see? You see a radioactively labeled DNA. And of course, this DNA had the genetic instructions to code for this bacteriophage protein code, so it allowed us to make more bacteriophages inside the bacterium. And this essentially confirmed this whole debate of DNA versus protein. Hey, DNA is the genetic material of all living things. So, Hershey and Chase, very simple experiment. This is nothing crazy, but hey, we haven't thought of it. They did. And this really sets a fundamental concept of molecular biology. So, alright, DNA is the genetic material of all living things. Great, easy. Let's talk about something else now. Let's talk about Chargaff. Chargaff was an interesting scientist. He essentially also discovered something very important to our understanding of DNA, this deoxyribonucleic acid. What a fascinating molecule, deoxyribonucleic acid. Oops. This DNA is very, very complex, very interesting, and over the years of scientific research, we've been able to characterize most of the structures and shapes and ideas of DNA and how it essentially works. And Chargaff said, okay, hey, I know there are 4 nitrogenous bases present within DNA. I know there are 4 nitrogenous bases present within DNA. So let's just name them. We have adenine, we have guanine, we have cytosine, and we also have thymine. Now, we're going to go back in a bit after we talk about the experiments. We're going to characterize and talk about all the characteristics of DNA, so it's going to be very simple, but let's look at this right now. Adenine, guanine, cytosine, and thymine, these are all nitrogenous bases. These are 4 of the bases that can be present on DNA. These essentially code for a protein. And what Chargaff realized was that the percentage of adenine present is also the same percentage of thymine present. So basically he said, hey, A is equal to C, and cytosine is also equal to guanine. So the percentage of cytosine is equal to guanine, the percentage of adenine is equal to the percentage of thymine. It's very interesting because now we're going to talk about, well, later on we're going to talk about Watson and Crick and Rowland and Franklin, and we're going to see how all this ties in together to help us understand about DNA. So, just remember this, A is equal to T, C is equal to G. Chargaff's rule. Now let's talk about Watson and Crick. I'm not going to go into too much detail about Watson and Crick, but all you have to realize is that Watson and Crick basically helped us realize that the structure of DNA is a double-stranded molecule. DNA is not single-stranded. DNA is indeed it's a double structure. So DNA is a double-stranded molecule. And of course, these guys have a mathematical concept, a mathematical model that essentially alluded to the idea that DNA has to be a double-stranded molecule. And Rosalind Franklin, another very, very famous and a very bright scientist, through X-ray crystallography, discovered that DNA was indeed double-stranded. And when Watson and Crick learned about our findings, they essentially said, hey, all right, our mathematical model has to be correct. And of course, they went on to publish their findings, and they were worth the Nobel Peace Prize. But that's besides the fact here. But the important thing to understand is that DNA is considered as a double-stranded molecule, and not only is it a double-stranded molecule, it also has a helical structure. These double-stranded molecules were being intertwined with each other, and it's a double-stranded molecule. And what holds these two molecules, these two strands together, are a bond noted as a hydrogen bond. So, both the strands are held together by hydrogen bonds. It's a double-stranded molecule. It has a helical structure. And Chargaff also said A is equal to T, C is equal to G. What they noticed was, hey, if we separate these strands, just to make our life easier, I'm not going to draw it all as a helical structure because then it's going to be a very messy drawing. But imagine this is actually an intertwined structure. So, hey, if I have a base here, a nitrogenous base, adenine, my complementary base is going to be a thymine, T. So here's the DNA base pairing rule. It's exactly Chargaff's rule. DNA base pairing rule. A is equal to T and C is equal to G. So therefore, if I have a G here, my complementary strand will have a C, or cytosine. If I have a T here, my complementary strand will have an A. If I have a C here, my complementary strand will have a G. Please memorize this rule. Adenine is complementary to thymine, and cytosine is complementary to guanine. Now, moving forward, let's talk about purines versus pyrimidines real fast, because this is also very important. So I've mentioned nitrogenous bases ATCG. And these ATCG fall under a specific category. They're either a purine or a pyrimidine. They're classified under two categories, so purine and pyrimidine. So purine versus pyrimidine. Purines have a 2-carbon ring structure. And the pyrimidine has a 1-carbon ring structure. Why do I care about this? Well, if on your exam you see a DNA strand and you happen to see a 2-carbon ring structure, a 2-carbon ring nitrogenous base, and the multiple choice asks you what possible nitrogenous base can this be, then you have to know what falls under a purine and what falls under pyrimidine. So let's look at it. I'll tell you right now, and then I'm going to give you an easy way to remember it. Purines, so adenine falls under the purine category, and guanine. So A and G both are considered purines. These are 2-carbon ring structures. You might say, hey, well, how do they look like? Well, they look like 2-carbon ring structures. Let's draw that over here. So here is my 2-carbon ring structure, and my pyrimidine looks something like this. Very simple, nothing fancy. And pyrimidine, well, if I have A and G here, well, the only ones left are cytosine and guanine. Cytosine and thymine. Cytosine and thymine are considered pyrimidines. And now you might be saying, wow, it's a lot of stuff to memorize. I have to memorize all this stuff, and now on top of that I have to memorize pyrimidines, 1-carbon ring structure, and so on and so on. Well, this might make your life a bit easier. Pyrimidine is spelled with the Y. Cytosine is spelled with the Y. Thymine is spelled with the Y. Y. It's a very, very easy trick to remember this pyrimidine function. Purine, well, whatever doesn't have a Y is a purine. Is that simple enough? It's a very easy way to remember pyrimidines. They both have Y's inside, and whatever doesn't have a Y, it falls under purine. So please remember that. That will help you understand, help you memorize very quickly. So okay, A and G fall under purine, and C and T fall under pyrimidine. I now want to take this a step further and understand, okay, A is equal to T, and C is equal to G. Therefore, when we pair A and T or C and G, that means that a purine will always be paired with a pyrimidine. Purine and pyrimidine. So purine does equal pyrimidine in this case. They have to have the same percentages, right? A is equal to T, C is equal to G. Notice one of them is a purine, the other one is a pyrimidine. Okay, very well. So now we've talked briefly about what's in Crick. We talked about Chargaff's rule. We talked about some of the characteristics of DNA. We're not done with the characterizing DNA yet. We're going to talk about that in another video. But now I want to talk about a very famous experiment that also sort of sparked the idea of biotechnology and molecular genetics. And this guy is called Fredrick Griffith. And this guy also gave very nice ideas in the genetic engineering field. Fredrick Griffith was a scientist who essentially discovered the idea of transformation. Now, you don't have to know what transformation is just yet. We're going to talk about transformation. But just remember, Griffith is the guy that is responsible for understanding or introducing the idea of transformation. We can understand at the end of this experiment what transformation is. This is what Griffith did. He said, okay, hey, I'm going to work with mice, and I'm going to work with certain bacterial strains. And he set up 4 different experiments, and he said, alright, I'm going to take mice. Here's my mice. I can't draw that well, so here's my very weird looking mice. And for the first experiment, he said, okay, I'm going to introduce a rough strain, naked bacteria, which is considered non-virulent. It's a non-violent strain of bacteria, meaning that it is not lethal to this mice. The mice's immune system can effectively fight off this naked bacteria strain, and it will still survive. And indeed, when you introduce this non-violent strain to the mice, this mice will happily survive, and all is well. Okay, experiment number 2, or setup number 2, I should say. Here's another mice. He's also happy now. Not for long, unfortunately. This time, he's going to introduce a smooth strain, encapsulated bacteria, and this is considered virulent. This bacteria has something surrounding it, protecting the bacteria from the immune system. So we call it an encapsulated bacteria, and it's a deadly strain. The immune system cannot effectively recognize, or rather fight off, this virulent strain, and this poor little mice will catch pneumonia, develop pneumonia, and essentially die. And that's exactly what happens, and at the end of the day, this mice is dead. So now he's sad, he's no longer living, he's dead. Alright, fair enough. If you introduce a violent strain, then, well, lethality is an option. Okay, great. Third setup. He said, okay, you know what? What happens if I introduce heat to this smooth strain encapsulated bacteria? By introducing heat, I should kill off this encapsulated bacteria, this extra layer of protection. I should be able to kill it off. And indeed, the protective coat around the bacteria is killed off. So this is a heat-introduced, killed encapsulated bacteria. And in this case, what do you think happened to this mice? Well, hopefully you've guessed that this mice is still surviving, right? Now that we've gotten rid of this protective coat, the immune system can once again now recognize this bacteria and fight it off and kill it. That is assuming that the bacteria is even active. So, great. A rough strain, non-violent strain, mice survives. This bacteria with an encapsulated layer kills the mice. If I introduce heat, this mice is still living. Here's where the experiment gets very, very interesting. This is what he does. Now he says, okay, what I'm going to do is I'm going to take a mice, I'm going to introduce the heat-introduced killed encapsulated bacteria, right? So the mice should not die, right? So he killed encapsulated bacteria. And I'm also going to introduce live naked bacteria. This is the rough strain, guys. Please, if you have forgotten that, this is the rough strain which was non-virulent. Rough strain. This is the smooth strain, but it's heat-killed. So, I'll have a heat-killed smooth strain which is virulent, but in this case the mice still survived. And I'm going to also introduce a live naked bacteria, which is a rough strain, but it's non-virulent. Remember that. In experiment 1, the non-virulent strain did not kill the mice. The mice's immune system was able to recognize this bacteria and kill it off and fight it off. Guess what happens to this mice? This mice ends up dying. This mice ends up catching pneumonia and dies. Why? Why is this the case? If I think about it logically, we didn't die here. The rough strain did not kill the mice in the first place in experiment 1 or in trial 1. Why is this mice dying now? And here's the idea of transformation. The idea is that this live naked bacteria, this non-virulent strain, was able to pick up some of the DNA, some of the genetic instructions for this encapsulated bacteria. And now this live naked bacteria has the genetic code, has the genetic instructions to make this encapsulated layer around it. And that's exactly what happened. This idea is transformation, picking up foreign DNA. In this case, we've picked up the genetic instructions, some of the genetic code to developing this extra protective layer. And all of a sudden, now you see this live naked bacteria producing this protective layer. And now it acts as a, what? It acts as a smooth strain. This is the idea of transformation, a very, very important concept. And we're going to talk a lot about this when we talk about genetic engineering and talk about E. coli and all this stuff. But just because we've begun about molecular genetics, I would like to get out all the experiments that I think are important out of the way. So, here's Griffith and his idea of transformation. In the next video, we're going to be talking about DNA replication. Before we do that, we're going to also talk about the characteristics. Stay tuned for part 2.